Okay, everyone. Thank you for being with us on today's webinar for Limb Girdle Muscular Dystrophy. And I'm excited today because this whole topic of genetic test reports has been an issue that so many of us have questions about. And we really haven't had a whole lot of educational resources in this particular area. So today we have Monkol Lek with us, and I'm going to introduce him formally in just a moment. And he's going to be doing part 2 of this series on demystifying genetic test reports for limb girdle muscular dystrophy, understanding boost. Now, understand we already did part 1, and that was already published on the Speak Foundation's channels. So if you're having difficulty getting to that, it's very easy to find. All you have to do is go to our website, and we have the most recent webinar published there. You just click on it, and it actually will take you to our channel, and you can actually watch all of the webinars that we've had up to this date. And you can also go back to the 2019 National LGMD Conference, and all of those sessions are published there. So you can get a whole bunch of information that you've not yet been able to see probably, and just watch all of those. The educational webinar series that we're doing will continue all throughout the year and into next year as well. And then in 2021, we will also do a national LGMD conference again, and that will actually be an international conference, and it'll be worldwide, and it'll be a virtual event conference. And we will actually advertise and present more about that next week. So I want to tell you about what's going on next week so you can kind of be aware. So next week we have something coming up where it's a simulcast webinar with Sarepta Therapeutics. So if you haven't heard about this, we're going to be doing a simulcast with Sarepta Therapeutics live. Their team will be coming on live, and they will be introducing a new limb girdle muscular dystrophy natural history study. That'll be next week on Friday, September 11th at 12 p.m. Eastern Standard Time. You can actually register for this simulcast today on our website. So if you go to the speakfoundation.com, and you'll see that on the front page, it is listed, and just register for it. And it'll say it's an Eventbrite. Just go right on there and register. It's a free event, and you're going to be able to hear from Sarepta's team, and they will also update you on everything. And also, I want to tell you that this webinar is sponsored by Sarepta Therapeutics. Sarepta Therapeutics has brought together the elements essential for development of gene therapies: productive collaborations, unparalleled scientific understanding, and an unwavering sense of purpose. Sarepta has 5 LGMD gene therapy programs in development, including subtypes 2E, 2D, 2C, 2B, 2L, and holds an option for a 6th program in 2A. It's important for you to realize there is a resource called limbgirdle.com. There, you can find out free genetic testing, where to go, how to get involved in that. There are free testing, 3 of them all throughout the United States, so you can actually sign up to do that. You can stay connected if you're a resident of the US and wish to stay updated on Sarepta's work. You just click on that website link there, and you can stay connected with them. And you can also connect with Sarepta's patient affairs team. So if you would like to talk with someone, you can contact them at advocacy@sarepta.com. So I'm excited now. We will have a Q&A after Monkol's slide deck presentation, but let me take a moment and go ahead and read you his bio. Monkol completed his PhD in medicine at the University of Sydney in 2012. Monkol is assistant professor at Yale University and has a lab located on the campus of Yale. He has been a senior research fellow for the Broad Institute at MIT and Harvard, and was a postdoctoral fellow at Harvard Medical School. Monkol is a lead analyst for the Exome Aggregation Consortium. Monkol has a strong passion for rare muscle disease research because he is also a patient with LGMD type 2G. Monkol is a member of Grasp LGMD. And at this time, I'm going to have Monkol come on, and he is going to share his slide deck with you. And then we'll do a Q&A with him. So go ahead, Monkol.
So thank you very much, Catherine, for the kind introduction. And thank you very much, everyone, for being here after a reschedule. So thank you very much for everyone. And also for giving up your Friday afternoon to hear more about the wonderful world of genetic testing and getting part two. So my collaborator and good colleague, Peter Kang, gave the first part of it, where I think he concentrated on the genetic report itself, the interpretation of the reports, and each one of the aspects. And part two will concentrate on when you get a variant of uncertain significance, or VUS, or VUS, or however you want to pronounce it. So first of all, I'll just introduce myself as a patient rather than as a researcher, because everyone's probably just heard about myself as a researcher. So as a patient, I've had quite a journey myself, trying to find a genetic diagnosis. So it took over 10 years to find a genetic diagnosis. And back then, genetic testing wasn't as rapid or as fast as now. But after receiving genetic diagnosis, one of the things that I wanted to do when I started my research career is to ensure that people wouldn't have to go on such a long journey to find a genetic diagnosis. In terms of what I did after that is I got a skin biopsy in 2016, because one of my collaborators at UMass Medical School had actually came out with an approach of actually trying to correct a base pair duplication that was causing my form of limb girdle 2G. And this is just some of the approaches they took. I'm not going to go into the details, but just to tell you, this is the journey I've been on, and and the importance of getting a genetic diagnosis, because it's the first step in actually trying to find a therapy for patients. But I'm going to touch again on genetic inheritance. I know Peter probably would have touched it on the three major forms of genetic inheritance that we look at when we go about producing the data for the genetic report and actually analyzing. The first is autosomal recessive. So this happens when your parents are carriers for the disease you have. So both have one defective copy. So you have, if you play out the mathematics, you can see here, you have a 1/4 chance of getting 2 defective copies of the gene, while you have about a 50% chance of being a carrier and only having 1 defective copy, but not have the disease, while you have a 25% chance of getting 2 normal copies and not even being a carrier. The other possibility is autosomal dominant, where, well, it's an autosomal dominant new mutation, or in other words, a de novo mutation. So neither of your parents carry the mutation that is actually causing your disease. And this is thought to happen when there is a mutation in either the sperm or the egg. So it happened on later, and then that got passed on. So typically you only get one affected in the family because of how rare this actually happens. And the last is autosomal dominant. So you have one parent with the disease, and so you have a 50% chance of actually passing on that defective gene. So you only have to have one defective copy of the gene to actually have the disease. So you have 50% chance of children having a disease. One of the challenges we have with autosomal is variable penetrance. So just because you have the mutation that causes the disease, not everyone's affected the same. And in fact, one of the most challenging scenarios is when the parent who is a carrier and should have the disease is very, very mild. And so it's difficult to detect, and and it can be mistaken for other scenarios where we don't know if the parent is on the mild end with the disease. So before I go into the details, I just want to touch upon the central dogma of genetics. So we have the DNA, and this gets copied or transcribed into a molecule called RNA, which is just a copy of the gene that has the instructions to actually make the protein. So we have DNA, RNA, and protein. So you have a mutation on the DNA level that impacts the RNA, but ultimately this impacts the protein. So you're either producing a protein, with less function, or it's not as functional, or sometimes you're not producing the protein, and this is what, you know, ultimately causes disease on a very high level. So when you receive a genetic report, this is just an example genetic report I got from Google Images, there are three broad scenarios that can happen. You get a definitive genetic diagnosis. So if you have a recessive disease, they're able to find both pathogenic variants. If it's a dominant disease, they're able to find the pathogenic variant and also confirm that the affected members in your family have it. So that's a definitive genetic diagnosis, and this happens about 50% of the time. But on the first pass, probably closer to 30% of the time, but eventually up to 50%. The other possibility is you don't get a definitive genetic diagnosis and you get a list of candidate variants. And amongst that list of candidate variants, maybe just one pathogenic variant when the genetic counselor was expecting two pathogenic variants because it's from a recessive disease. Or you get two variants of uncertain significance or a pathogenic variant and uncertain significance. I'll step through it on the next slide. And the next possibility, which happens to about up to 50% or even more, is that there's no candidate variants found. So out of all the variants that they found, combing through it, they were unable to find anything. And this is what my other area of research that I'm not presenting, it concentrates on what do you do when you actually have absolutely no candidates. And so one of the reasons why is that it's a novel disease gene that's causing your disease, and it was unable to be either interpreted in the genetic report or detected in the genetic report. So in terms of the candidate variants that can be reported, like I touched upon, it could be a variant of uncertain significance. It could be just one pathogenic variant, or it could be combinations of above. But one thing I do want to highlight is that if you have candidate variants in multiple disease genes, it does not mean you have multiple diseases. I've had patients before come to me during patient conferences and show their genetic report and say, oh, do I have this disease and this disease? I'm going, well, they are candidate variants. And it's very, very rare that someone actually has two neuromuscular diseases. I'm not saying it's impossible, but I'm just saying it's very rare, very rare. Listing candidate variants is just a way of just being comprehensive in the listing. And once one of them becomes a clear candidate based on other forms of evidence or information, that is typically the variant, and the other candidate variants typically then become likely benign or stay as variants of uncertain significance, knowing that it probably doesn't cause the disease. So going in variants of uncertain significance, so these are variants in disease genes. If I put it in my word, is that the geneticist or the genetic counselor that went through and interpret the variants, they're just unsure if it causes a disease. And I just have to highlight it again, they're unsure if it causes disease. Some people have seen that and have misinterpreted and have said, they've just seen it's on the first page and say, well, I have that disease. It's most of the times it means it's unsure. But what actually tips sometimes neurologists and geneticists over is if it comes in combination with a pathogenic variant, but still it's the variant in isolation itself, we're just unsure if it actually causes disease. And one of the things I didn't get an appreciation of when I was a patient before I became a researcher is, why is it so difficult to know if they can cause disease? So in my research, we performed this thing called exome sequencing, where we sequence, we attempt to sequence all known genes in the human genome. So that's about 20,000 genes, where just under 20,000, we actually do get good coverage and capture of. And what you find when you sequence that you have about 20,000 variants. So what that means is 20,000 places where your DNA in your genes are different to people in the general population. It doesn't mean it's different to everyone, but it just means it's different to some people. And amongst those 20,000, 12,000 of them are likely to impact protein function. They can impact protein function in two different ways. One, like I said on the slide talking about the central dogma of genetics, is Some of them, which we call loss of function variants, result in no protein at all. And you can see why it may cause disease. If you're not producing enough of that protein or not producing that protein at all, that can cause disease. The other possibility is a missense variant. So that impacts on the function of the gene. The gene has to do a particular function in the cell and it can't do it, or it can't do it well. Again, that could lead to disease. So there's 12,000 of those variants that could be good candidates. But amongst the 12,000, only about 100 to 500 of those are what we consider rare in the population. And the reason why we're only interested in the rare ones, so that means it's not found in many people, is that each person with limb girdle muscular dystrophy or any other rare disease, it's a rare disease. So what it means to be a rare disease is that what that means is that there is not many people in the population in it. So that means the genetic cause of your rare disease, it shouldn't be that common in the population, or there should be more people in the population with it. It's just a logical argument. So some of these will be disease genes associated with patient disease, but it doesn't mean it causes disease. So one of the things that, you know, it's hard when I was a patient to grasp this, but rare genetic diseases are caused by rare variants, but rare variants do not necessarily have to cause rare diseases, because if you take a healthy person, you will also find 100 to 500 rare variants, and they don't have disease. And some of these rare variants actually happen in muscle genes that we know cause disease, such as titin, nebulin. So these large genes, just by chance, will have these rare variants. And they're found in perfectly healthy people. So when we see a rare variant now in a patient, we can't automatically conclude that it causes disease. And so that's the challenge in interpreting rare variants. And a lot of these are actually interpreted as variants of uncertain significance. So just a snapshot of how difficult the problem is. This is the challenge of interpretation. So this is taking a snapshot of the Jain Foundation cohort, where they sequence approximately 2,500 patients with limb girdle muscular dystrophy, with the 30 or so genes known to be associated with limb girdle muscular dystrophy. And the variants of uncertain significance here is colored in blue, and can see down that breakdown where I'm showing in red pathogenic and likely pathogenic. And you can see the larger the gene, the more likely a lot of the variants will be interpreted as variants of uncertain significance. So now I'm going to go into the 4 main ways we can actually resolve a variant of uncertain significance. So be able to tell if it's likely benign or likely pathogenic. Like most of the times we're normally ruling it out rather than ruling it in. So the first of these is segregation. So and a lot of this is in the context of recessive diseases. So 2 variants can occur across 2 copies of genes in the following ways. So if you can think of these as the 2 copies of genes, one possibility is you have a mutation on one copy and a mutation on another. So overall, you have 2 defective copies. The other scenario is you have both mutations on the same copy, while the other copy has no mutation. So you only have 1 defective copy. So this is less likely. Well, this is unlikely to cause disease, because you need, in a recessive situation, 2 defective copies. But when you only sequence the patient, and this is typically the case when it's limb girdle patients, because they're adults and they normally don't go to the clinic, well, hopefully don't go to the clinic with their parents. I never went to the clinic with my parents. So it's much harder. You have to get the parents in, the family in, to do sequencing on the whole family. So what we're expecting when we do sequence the parents, so this is the only way we can tell between these 2 scenarios, is to actually sequence the parents. Because you can just think of this copy here coming from mum and this copy here coming from dad. Again here, 1 from mum, 1 from dad. So if you can sequence mum and dad, what you're looking for is both, each one of them being a carrier of one of the pathogenic variants that are actually causing disease, rather than one parent being the carrier of both the variants. So that's what we expect. The parents to be carriers, that is the carriers of one candidate variant of the disease gene. But the alternate that could actually happen is one parent has both these variants. So when one parent has both the variants, it's very unlikely that both of those variants are disease causing, and they're normally classified as benign. So it's a way of actually not ruling in a variant of uncertain significance being pathogenic or disease causing, but rather ruling out by elimination. So in the case of de novo dominant, in this case, we really need to sequence the parents. And actually, you can't actually come up with this conclusion unless you actually sequence the parent, because if you just detected a heterozygous variant, so just one copy of that variant on one of the genes, you would not know if it was de novo unless you actually sequence the parents to say, we don't observe it in the parents. But what tends to happen, a lot of these sequencing data is from next generation sequencing. So what tends to happen is when it doesn't validate is sequencing error. So this typically happens with research sequencing and not clinical sequencing, because research sequencing is not at high enough coverage to actually tell the difference. So sequencing error is normally underlying when they don't validate, but that will typically not happen with clinical sequencing. This is more something that happens on a research basis. And the last the last scenario is resolving the variant of uncertain significance in the case of autosomal dominant. So here what we're expecting is whether one of the parents is obviously affected or not, that they carry that variant that we believe is actually causing the dominant disease. So for example, the dominant form of limb girdle muscular dystrophy. So the alternate that could happen is neither of the parents carry the variant. So if neither parents carry the variant, it's very unlikely that this variant causes disease. So again, we're ruling out. So this is a thing we do a lot for dominant diseases, just ruling out, not just with the parents and seeing if they have the variant, but also the family members themselves and if they have the variant. So, you know, there's typically, because there's a 50% chance, so that sometimes in a larger family, so 2 generations, 3 generations, 4 generations, that we have multiple effectors that we can rule in or rule out. But one of the challenges of ruling in, ruling out is that a lot of the dominant variants have variable penetrance and severity. So sometimes it's not obvious if they have the disease, and sometimes it may be onset later on in life. So although they don't have the disease right now, they may get the disease later on. So it's very hard to make what we call a genotype-phenotype correlation sometimes with these dominant diseases. So the next way we can actually resolve a variant of uncertain significance, and this is probably the most common way of resolving it. A lot of times when a variant is considered pathogenic is because it's been observed in other patients before. So typically unrelated patients. So not, you know, someone that's closely related, a cousin, you know, an uncle or auntie, but, you know, someone that's just totally unrelated. So one way you can actually look at that is looking at mutation databases. So typical ones are the Human Gene Mutation Database, which is actually a commercial database now owned by QIAGEN. So it's not as accessible, but 2 accessible databases are the Leiden database, maintained in Leiden in the Netherlands. They have this open database where you can look at variants, and also the NIH database, which is the Clin database, where you can go to the website, look to see if someone has your variant and whether it's been reported before. So it only has high-level data to protect patient privacy. So, but the great thing about the Clin databases, and so also the Leiden, the Human Genome Mutation databases, is they also associate it with publications. So it's a good way as a patient to actually read more about the pathogenic variant that's causing your disease or that's running in your family. So the report typically looks like this. So I've taken a variant from FKRP, and here are all the details. So it has the genomic coordinates of the variant, the classification, the star rating, oh, the classifications here, and also who submitted that report. So I think, you know, in terms of getting pathogenic variants, this is the most likely way of classifying. And it's also the most likely way over time, because this information is not static. We're constantly learning and we're constantly finding other patients with the same genetic variant and the same disease. And so it may be classified as variants of uncertain significance today, but maybe in 2 years' time, when they've gathered more evidence or found another patient who has the exact same disease, that classification can actually change too. And it also can change the other way around. So one of the things that we did in one of my research projects, the Exome Aggregation Consortium, is show that although a variant is very rare amongst the Europeans, it was quite common amongst Latino population and too high to actually cause a rare disease. And we not only did that, we actually found the carriers of that rare disease and actually checked their liver function, because they were supposed to have a liver disease, and showed they were perfectly healthy. So either the variant has a variable penetrance and doesn't always cause the disease, or that it wasn't actually a disease-causing variant. So it can go either way, the variant of uncertain significance. It can become likely benign, or it could be likely pathogenic. So the mutation database, even though it's great, it typically only works well for common or founder mutations. So like I said, you know, the assumption is, you know, if it is common or common enough that there is another person in the world with the same mutation as you, then it may appear there. So it works very well for calpain-3, FKRP, and NO5. So all common forms of limb girdle muscular dystrophy, where a lot of patients actually have a common founder mutation. But one thing to note is the remainder pathogenic variants are ultra rare and generally not observed in other patients, or very few patients. And so then you've got to think about the probability of that person being discovered, you know, in a genetic study where they're actually reporting to one of these mutation databases. And so, you know, those two things happening is very unlikely. So, you know, not only in the space of neuromuscular diseases, but other diseases like cardiomyopathies and other diseases, they found out that it's unlikely to resolve a variant of uncertain significance. And typically the ones that have been resolved have already been classified as pathogenic, likely pathogenic, but it could resolve it over time as we keep continuing to accumulate data here. So a traditional way, so they're the two most common ways of resolving a variant of uncertain significance, because it takes the least time and money. So the next one is a lot of patients in the audience and listening to the recording go to neurologists or doctors that actually have research labs, so physician scientists, and a lot of them actually look at functional assays to have a look at, does this variant cause disease? And one way we can look at is protein levels. So some variants impact the protein level or the protein size. So I'm just showing you FKRP, where these mutations have impacted protein level. This is a Western blot where the black represents the amount of the protein here. And it also can impact the protein size. So you can see this band here is lower than the other band, showing that this mutation here actually also impacts the size of the FKRP protein. The other thing that can happen is protein localization. So this is an example of the cycloglycans, where cycloglycans, the healthy wild type, in this case example I'm showing you, alpha cycloglycan, is found at the cell membrane here, colored in red. And what happens when there is certain mutations in cycloglycan, this affects its ability to get to the membrane, where it actually does its function, does its job at the membrane. So this is another way of actually doing. Again, each one of these are very laborious to do one mutation at a time. Another way, and this is actually, this is a shameless plug, this is my wife, Angela, looking at dysferlin and its ability to get to the cell membrane. So when you create muscle damage. So here she's used a ballistic assay where she's firing gold beads at muscle cells, creating a hole and seeing how quickly proteins involved in membrane repair get to the area of muscle damage, where if there is a mutation, it is defective and doesn't get there as fast, or it doesn't make such a tight ring around the mutation. So these are examples of ways we can actually look at it, but each one of these have to be physiologically relevant also. So cycloglycan has important function at the cell membrane. So it makes sense that if it can't get the cell membrane, it's very likely to cause disease. In this case here, in this membrane repair assay, genes just such as dysferlin, known to play a strong role in membrane repair, is expected to go to the site of damage. So the last thing is high throughput functional assay. So going back to that concept of just because a variant is rare, it doesn't mean it causes disease. And also the concept that I just showed last slide, a lot of this work for each mutation, this could be 3 to 6 months worth of work, if not even more. So if we did this for every patient mutation that was considered a variant of uncertain significance, it's not going to scale. So I would say that when I first started my PhD, and a little bit before my PhD, the saying was one patient mutation, one PhD. Unfortunately, that sort of like that framework is not going to scale anymore, given the amount of genetic testing we have and the amount of variants of uncertain significance. So the idea in the last thing of how we can resolve variants of uncertain significance is with high throughput functional assays. So to find a functional assay that we can rapidly test things. So I'm not going to go into the details of that, but the idea behind this, and this is research that my lab's doing funded by the Muscular Dystrophy Association, is instead of waiting for a patient, doing a functional assay on their mutation, the idea is let's not wait for the patient to come. Let's create every possible mutation in the gene and then push it through a high throughput functional assay, which you could do in one afternoon and have a readout for every one of those mutations. So it becomes a lookup table. So a patient comes in, we see a variant, it's classified a variant of uncertain significant, and then we look up in, so what signal did it produce in our functional assay? And therefore, it will help us either push it towards likely benign or likely pathogenic. So it is not the piece of evidence that allows us to go either way, but what I consider another line of evidence to either support going either way, because this is very important for patients to actually get a genetic diagnosis. So this is one way in terms of research and active research we're going to address it, because this is a more scalable way of addressing it. So just to wrap up, I've touched upon 4 ways we can go about resolving variants of uncertain significance. The first is segregation. So each one of these require follow-up after genetic report. So segregation, so sequencing particularly both parents to see does it segregate correctly. So with limb girdle muscular dystrophy, a lot of the research studies I'm involved in, we consent the patient, but we typically don't get the DNA from the parents. So to get a definitive diagnosis, you really need that to tell the difference of both the pathogenic variants in one copy of the gene, or is there one on each copy? Because that is the scenario we're looking for recessive diseases. So, the gold standard is to sequence both the parents. The other thing I touched upon is looking mutation databases. And this is typically to give you classification of the pathogenic variant. But what it will give you also looking into the future is, as more data is gained through these mutation databases, there'll be more and more patients, with reports, with your phenotype, with reports of that VUS or VOUS. And then that, it will reach a threshold going, that's probably a pathogenic variant, because we got multiple unrelated patients around the world carrying this VUS. And, a geneticist will make that call after a while, after that accumulated. The third thing I touched upon is functional assays, as I showed you the different functional assays. And the last thing is, how can we do that in a high throughput manner? So not wait for every patient mutation to come in one at a time, but generate data for every possible mutation in a patient. So I'm going to end off why it's important to receive genetic diagnosis, because as a patient in my 20s, I thought, well, if there's no cure, well, why bother? Well, there are other things other than just having a cure. The first is lifestyle choices and preventions. A lot of these genetic diseases, there is a genotype-phenotype correlation of how the disease will progress. If you know how the disease is going to progress, sometimes you just have to accept and be mature and say, okay, while I'm waiting for better treatment, how can I take better care of my body? So if you know you're going to have issues with your heart, issues with this, issues with that, then you can manage it before it becomes an even bigger issue. Next is available treatment for misdiagnosed patients. So what we tend to find with our limb girdle patients is not all of them actually have a genetic limb girdle muscular dystrophy. So some of them have myasthenia gravis, which is an autoimmune disease, which look like limb girdle muscular dystrophy, where there are drugs that actually treat them. So I just was chatting to one of the patients who also happens to be a scientist. I'll keep the details private, but he shared with me 2 weeks ago, his decade-long journey was over and he didn't even have a genetic form of limb girdle muscular dystrophy. So all these years, his neurologist thought he had limb girdle muscular dystrophy, but he has an autoimmune disease, and he was immediately put on the right medication and he started seeing his CK levels drop. So that's another reason why you should keep pushing, whether it's a genetic diagnosis or a diagnosis in general, because you may not even have what you think you have. Unless it's definitive, it will always be up in the air. The next one is eligibility to enroll in some clinical trials. As Catherine had mentioned at the start of the talk, Sarepta has some gene therapies for some of the forms of the sarcoglycanopathies. So, but to be able to enroll them, you have to have a definitive genetic diagnosis, not just a diagnosis where a doctor thinks you have a particular form of the disease, but associated with a genetic mutation, because a lot of therapies now are very specific to the gene. And if you're not sure if it's that gene, then it's not a good use of resources to apply a particular therapy, especially if it is a very expensive therapy. The last is the design of rational therapy. So I think we're living in a very exciting time of personalized medicine. And so this opens ideas of, if we know the exact mutation that's causing your disease, there are many different approaches now we can take in patient cells and also mouse models where we've engineered the patient's genetic mutation that we can actually test. But knowing the root cause, or the genetic root cause that causes your disease, we can't actually explore those options. So hopefully I've, convinced people in the audience and people watching this recording why it's important to receive a definitive genetic diagnosis, and why to keep pushing on when you have a variant of uncertain significance and what I call, you're in genetic purgatory. But and with that, I'd like to wrap up and take any questions, and I'd like to thank Catherine again for giving me this opportunity to share with you just my thoughts of variants of uncertain significance and how we can go about addressing them.
Great. Thank you so much, Monkol. So what we're going to do at this point is have a Q&A, and we will have so many questions here. As you were going through your presentation, I got to say, excellent job. There were so many things that you talked about that were questions I had, and then it just brought up more questions, and I thought, my gosh, I'm going to drive Monkol crazy if I bring all this up, because I have so many questions. It's just awesome. It was really good. We had some people submit questions in advance. One of the questions, and I'm going to read it, and it is a little bit more detailed, so I know you're going to follow with me. One person submitted this question. My genetic test revealed one pathogenic mutation and one boost. Based on my symptoms, I was diagnosed with LGMD2L. I used the Clin website, and I see that one of the mutations in my ANO5 gene is pathogenic, and one is of uncertain significance. I'd like to better understand the review status column for the variant of unknown significance. It reads, criteria provided, multiple submitters, no conflicts. What does this mean? And does this indicate that they may be closer to changing the status of that variant to pathogenic?
I think it's a fantastic question, because I think it's a very typical question for patients with FUS. It's a great place to, it's a great resource. As I mentioned during my talk, ClinVar, it's a NIH government supported resource, and it's free for patients to access also. So I like that this patient's very savvy and actually accessed it. So I'll go over, so criteria provided. So in order to actually submit a variant interpretation, a clinical variant interpretation, companies and also academic institutes have to say what their criteria for classification. So the classification of pathogenic, likely pathogenic, benign, all those classifications was set by the American College of Medical, I think, Genetics and Genomicists. But I'm not 100% sure on the G, but you can look it up yourself. So they set guidelines on how to interpret and classify these variants, but within these guidelines, there's still some room for interpretation. And so each company and institute will say, this is how we've interpreted some of the things that were more open to interpretation. So that would be the criteria provided. And based on that criteria and how detailed it is, submitters will get a star rating going. This company, has fully detailed everything, and therefore this is a 2-star or 3-star rating. And so that's what it means by that. Multiple submitters is good, because that means that it's seen by multiple companies or multiple submitters, academic institutes and things like that. And no conflicts is another good thing. So a lot of times there are conflicts. It's unfortunate that the guidelines are open to interpretation. So some groups may classify it as variants of uncertain significance, while another group may classify it as a likely benign. So it's good there's no, but one caveat I got to let the patients know that a lot of patients have very, very rare variants. You may be the only person in the United States with that. So sometimes, probably not in this case because there's multiple submitters, unless that patient went to got their genetic testing at multiple places. A lot of times that one submission is actually that patient themselves. So when I said that, it's a way of finding another patient, but a lot of times that, if you see the company listed and you realize that that was the company you got the genetic testing from, it's most likely you.
Okay.
I know, it's a weird thought, but, and it's one of the challenges we have with, say, natural history studies, patient registries and things like that. We want to have all the information. We want to maintain privacy. So, I'm on the MDA MOVA patient registry, and I'm saying, you can't have it all. You can't have privacy, but have the variant, because that patient may be the only person there with that variant. But the other point I'll make to that question is, if they're getting closer, well, I think they are. I'm hoping that, if there are multiple submitters and all matching to the same phenotype, so the same limb girdle subtype, that the next time a company submits, they'll go, well, we've had like 5 variants of uncertain significance. We know that this isn't from the same patient going to 5 different companies, that maybe we'll put it as likely pathogenic, but put the reason why. And then what will happen is a lot of genetic companies such as GeneDx have this annual review or a biannual review where they'll look at all their patients that were classified as variants of uncertain significance and go, based on how mutation databases have changed, does the interpretation for this patient change? So rest assured for that patient, if it went through one of these genetic testing companies, they have made it a habit to make this review. Companies have made sort of like drawn a, you know, cost to benefit ratio of, you know, should we do it every year? Should we do it every 6 months or every 2 years, based on how often data actually changes?
That's
I know
good.
it's a long-winded way of answering the question, but that's what goes into considerations.
No, that's very good. Do you feel that because she was diagnosed already with 2L, so I know in FKRP, for example, I have homozygous 2I, a 276, is what I always say in my head. So I know a lot of people with 2I have a heterozygous form. So it typically, what we see is the homozygous form is a milder phenotype than the heterozygous. So would that follow similarly with something like 2L? So if they got the general mutation that is associated with 2L, but then they have this variant, would that, if they find that it's disease causing later on, will that like modify the phenotype to be more severe, or not necessarily?
Not necessarily. We're still trying to understand sort of like the genetics of, you know, variable, sort of like variable severity and things like that. But it's a good point that you brought up. What we tend to find is that the more common ones, when we do find in homozygous state, like the N05, the common variant, the common pathogenic variant in N05, is that it's typically later onset and less severe. And it kind of makes sense in terms of human genetics, in terms of that's how it got to a higher frequency, because it's less severe. So people are more likely to get to the age where they can have children and the variant get passed on. While more severe forms, you're unlikely to get to an age, you know, and or have a lot of children.
Okay. And then when we say homozygous too, I just wanted to make sure everybody understood. So that meant that the disease came from on the mother and the father side, that it's the exact same mutation. And that's correct, right?
Yeah, but just to correct you on the other point, typically what we use the term compound heterozygous, so it's two heterozygous variants, right? And if you use the word just heterozygous, it normally just implies one.
Okay, great. I'm glad you're sharing that.
Yeah.
Now, here's another question that was submitted. I guess in the world, COVID's such an issue, of course, and we're all looking for safe ways. And one person asked, is there a way for them to find a genetic test for their subtype via home? So I guess they were wondering, can somebody send them like a genetic test at their home? Can they do like a cheek swab or a saliva test, versus going in and doing any blood work at like Quest Diagnostics or something similar?
Yeah, so most definitely. So there are generally three ways you can extract DNA. The gold standard is blood, but I'll go back, touch upon that. There's saliva or there's cheek swab. So our lab tends to find, yes, blood's the best. The next best thing is saliva. The next best thing is a cheek swab. But the great thing about some of the research studies that we do, we just send a cheek swab. They're the cheapest kits. I think they're $1 to send a cotton bud to people to their home so they can swab themselves, or a saliva kit, you know, build up as much spit as you can and get it to the line. So the DNA extract from that, although it's not the gold standard, it is more than sufficient to do panel sequencing, like the Jain Foundation panel that I presented, or panels that Council does and Invitae does, and also more than sufficient for exome sequencing. What it's not sufficient is for other tests like genome sequencing and other clinical tests, but it's typically good enough for a lot of the things that limb girdle patients need. So I know, you know, we're in the period of COVID and things like that, and I highly suggest for people to do that initially. There isn't a need to actually go in and give a blood draw.
Okay, that's good. What's your thought on muscle biopsies to diagnose limb girdle? unnecessary?
Well, I had a muscle biopsy, and you got to excuse my language. It bloody hurt. It hurt for 3 days. This is not discouraging people to get it, but you know, it depends what your goal is. If your goal is to get a genetic diagnosis, I think the first line now generally is to actually just give saliva for a genetic test. What we tend to now use biopsies in the context of research is either one, a way of resolving VUS, so we can look at protein levels from the muscle biopsy. But in terms of translational stuff, like I showed on my first slide, it's a way of creating cells from that patient and try different genetic correction strategies. So it's a really great tool for research. I know it causes a lot of pain because I've been on that end of pain, but you know, earlier when genetic testing wasn't so great, it was a form of getting to the right gene by looking at the protein levels, but we don't use it as much. So it depends on the goal of the patient.
Yeah, I tell you, the muscle biopsies have really changed over the course of 25 years. My first one was an open biopsy and a 4-inch scar. And over the years, I've had, I think, 6 now. They are now to the point it's like a little pinprick. So if you have to get one now, they're really not as horrible as they were before. It actually is manageable, and it's a very small little scar. And I have to laugh because the technology from 25 years ago to now is like completely different. And but for genetic diagnosis, you know, I would agree with you too that if someone's going to a rural place and they say you need a muscle biopsy for a genetic diagnosis and they have limb girdle symptoms, that's probably not the best first line, right?
Yes, I agree. But thanks for correcting me, because yeah, muscle biopsy technologies are a lot better than when I got it. I have that same large scar, and you know, and you're a much braver person than me. The amount of muscle biopsies you're given, and you know, I think researchers are very thankful for that too, because it does help us, you know, understand the disease better.
It is. It really is helpful, and especially if you're going to get any kind of drug treatment in the future. You know, a muscle biopsy is probably going to be required to be able to show the protein in the cell, probably. So, you know, just saying that as a caveat. But you know, because they're so much better than they were, they're, you know, just, it's really just a nuisance more than anything, I think. Okay, and then I was going to ask you another question. How can a person find out about current clinical trials that are going on right now?
I think there's 2 ways. So one is there's a great website called clinicaltrials.gov. So you can go to that website. So for example, the Sarepta gene therapy trials are on there. A lot of the mini dystrophin, so the one done by Solid and the one done by Pfizer's there. They have numbers like ID numbers and things like that. So that's one way as a patient. It's very quick, like the patient that went to the Clin website. It's open, it's public, you can see. And you can see also a lot of them put their inclusion criteria also. But, you know, I'd give them a call anyway if you didn't fit the inclusion criteria. So that's one way you can help yourself. The other way is, one way, it sounds funny, is being involved in the patient community also. So word of mouth or hearing from other patients. Sometimes I hear about them through that. And the last is your neurologist. So, you know, I hate to say this, but I feel like if you're going to a neurologist that is unaware of clinical trials for your disease, maybe you're seeing the wrong neurologist. So, you know, it's worth it to just travel that extra, say, 3 hours once every 6 months or a year, because you want to have a neurologist that's switched on, you know, and not a neurologist that just writes notes about your disease every 6 months and says, I'll see you in another 6 months, but one that says, well, I know this clinical trial, you know, some of them are small molecule ones and things like that, but you want to know and have that option. And normally the neurologist knows, they probably know the person running the clinical trial too. So you have an in.
That's such a good point. And, you know, I think about the Grasp LGMD Consortium, that all these educational webinars are kind of partnering with Grasp. And what I'm so appreciative of is the fact that there is a consortium now of neurologists that are getting together and discussing research, and that you guys are all aware of all the clinical trials going on and developing them as researchers and doctors. And what a great point you mentioned. How important it is if you know that there's a particular doctor in the United States that specializes in your subtype, that you contact their office and see if they have any natural history studies that you can participate in. Oftentimes there's, they actually have funding to actually help you with travel costs. So it is really important. I started seeing Dr. Matthews in Iowa about 12 years ago. And I first started there because of a clinical trial. She was doing a natural history study. And I think that that was really good. Now, all throughout my life, I've had great neurologists. So it wasn't a case of not having a great neurologist. It was just she had like, she was specializing in 2I and was doing a natural history study. So I knew that she would see tons of patients with my form. And it really does help because she's going to tell you about clinical trials. And you hit the nail on the head. You know, my doctors have always been telling me about clinical trials. You're eligible for this, or you ought to think about this. And then just being in the disease community, like you mentioned, and talking to different patients, sometimes that in and of itself will help you because they'll tell you about a trial that they're in if you didn't know about it. And looking at the website, clinicaltrials.gov, all the time. And that's so important. I was also going to share that we'll be doing the simulcast next week with Sarepta, and they're introducing that new natural history study. And then in October, we're actually doing a webinar with ML Bio, and they're going to be talking about their clinical trial that is going to go on, and that is going on right now. And that's for 2I. So there's a lot of things that are coming around the corner that people really need to make sure that they're looking into, because they're, like you said, knowing your genetic diagnosis means that you might be eligible to do a trial. And that's very, very important. I think you're going to be interested too. On the 18th of September, we actually have a CRISPR webinar, and it'll be on 2G and 2I. So the guys out of UMass will be doing that, and that's sponsored by Cure LGBD2I. So I was going to tell you about that one. If you're interested.
So that must be Scott Wolf, right?
It is. It sure is.
Yes, yes. So he recently gave a presentation at the MDA conference also. My wife was the chair of that presentation.
There you go. Yeah, he's excellent. So we're so excited. Yeah.
Can I reinforce one of the points that you made, Catherine? And that was, I'm guessing you don't live that close to Kathy Matthews, you're saying, and can you like reinforce what I'm trying to say to patients is, yes, you do have to travel quite a bit, but to have a doctor who's with Kathy Matthews is actually leading the clinical trials, not even, you know, not let alone knowing about it, but she's leading it, that gives you sort of like, I think, you know, the best possible care.
Oh, absolutely. You know, that it is, you know, a difficulty because I live in Georgia to travel to Iowa. I mean, it's not an easy jaunt, but it is one of those things that I am so thankful about, because she was the one that made sure that my breathing was watched every year. And I have had PFTs every year for the last 12 years, echocardiograms every single year. I'm just followed very, very closely. So in case something comes up, you mentioned it, you know, we are already looking at it. So if it starts to emerge, we can address it early on. And there are some serious conditions that come with some of our limb girdles that can affect our breathing. We can have cardiomyopathy. So making sure that we don't have those things. And if we start to see them, we address them early. I've had 2 cardiac MRIs in the last 7 years, and I've had echoes every year. Well, my MRIs show no fibrotic tissue, but you can't see that on an echocardiogram, actually, to that detail. So just knowing this kind of information is so important. And yeah, travel, if you are able to, please do. It's very, very important for your health, I think. Yep. All right, so at this point, we're going to go ahead and close our webinar with Monkol. And Monkol, I just want to say what a great job you did. I'm so thankful. Sorry for all that you had to go through last week with that tornado, and we're just so glad you lived through it and that you were able to be with us today. So thank you very much.
Yeah, it was fantastic to spend time with the patients, family, and and just interested researchers that are on this call. And
Yes.
thank you very much for the invite. It was fantastic to put this together and present.
Awesome. Well, we will publish this professionally to our channels and our platforms in the next few days, and then you'll be able to watch it or rewatch it as you need to. And we want to thank you so much, and may everybody have a safe holiday this weekend. Bye-bye.